Okay, so this is the recording for the second part of chapter 3, which is biomolecules. So we're gonna start off talking specifically about carbohydrates, and we will talk about each class of biomolecules. All right, so in terms of functions, right? Functions of carbohydrates, they are listed here, right, as an energy source. Also as providing building materials, right, a structural role. And so we'll talk a little bit more about this as we move through. But basically, glucose is a carbohydrate, right? That is the energy source of your cells, right? Your cells break down glucose basically to generate ATP, okay? And then ATP is the energy unit of the cell. Building material structural role. I kind of mentioned that in part 1, that there are structural carbohydrates that provide structure to plant cell walls. Well, again, I'll mention this, and this will come up again as we move through. Carbohydrates contain carbon, hydrogen, and oxygen in a 1:2:1 ratio, right? So 1 carbon, 2 hydrogens to 1 oxygen, okay? There are different varieties, monosaccharides, remember mono meaning 1, disaccharides 2, and also polysaccharides, which are many, okay? Monosaccharide is a single sugar molecule. Remember we mentioned monosaccharides before as being the subunits of carbohydrates or the monomers, okay? Monosaccharides, one single sugar molecule. They're also called simple sugars, and they have a backbone of about 3 to 7 carbon atoms, okay? Biggest example, glucose, right? Fructose, right? You find in fruit. Galactose, those are also examples of simple sugars or monosaccharides, okay? These guys here, these examples are all hexoses. They have 6 carbon atoms. The sugars ribose and deoxyribose, which you find in DNA, ribose in RNA, deoxyribose in DNA, those are 5 carbon sugars or pentoses, okay? So this is basically just showing you glucose, right? We already saw this before, C6H12O6, right? Remember in that ratio 1:2, right? 1:2:1, okay? And just showing you different ways that you can represent glucose. Do you need to draw the structure of glucose? No. Am I going to ask you to identify glucose based on one of these structures? No. Should we recognize this as glucose? Yes. Okay. All right. So glucose monosaccharide. Okay. We also have disaccharides, like we mentioned. So these are 2 monosaccharides joined together. And remember, they're joined together by dehydration synthesis, right? Or a dehydration reaction, like we talked about before. Some examples of disaccharides, lactose, right? Which we find in milk. It's composed of galactose and glucose, right? So lactose is composed of these 2 monosaccharides. Sucrose, right, table sugar, is composed of glucose and fructose. Maltose is 2 glucose molecules joined together, okay? So these are all examples of disaccharides because you have 2 monosaccharides joined together. All right, so like I said, in part 1, we're going to constantly come back to these synthesis and these degradation reactions that are specific to each class of biomolecule, right? So we talked about them in general, and now we're going to talk about them again and again each time we talk about a different group of biomolecules. Okay, so for carbohydrates, our example here is showing you the synthesis and the degradation, right, or breakdown of maltose. Remember, maltose is a disaccharide that's composed of 2 glucoses, right? Okay, and you can see that here. So you have 1 glucose, you got another glucose, and we're going to join them together, okay? And so we join things together. We join glucoses together by a dehydration reaction, right? Synthesis is a dehydration reaction. And so now you join these 2 glucoses together and you formed maltose. Remember, dehydration, you lose water in the process, all right? If we go, so there you go, showing you that 2 monosaccharides joined together to form a disaccharide. And again, you're losing water in the process here. This is a dehydration reaction. If we go the other way, right? And we're going to break down maltose. So we're going to break down our disaccharide. We're going to break this bond right here. Okay. You're putting water into this reaction, right? We're following that arrow. It's a hydrolysis reaction. Okay. What happens here? Again, hydrolysis, this is degradation or breakdown. We had our maltose, and now we're back to our 2 glucose subunits here. Okay? Hopefully that's clear. We should understand this whole process here. All right. So now let's talk about more complex carbohydrates called polysaccharides. So it says a polysaccharide is a polymer of monosaccharides. So now polysaccharides have many, many monosaccharides joined together. We just mentioned disaccharides, which are only 2. These have many monosaccharides. Okay. So it says energy storage. So for the most part, when we think about these complex carbohydrates, these polysaccharides, it's for energy storage. So in plants, the complex carbohydrate or the polysaccharide that stores energy is called starch. Okay, so energy storage in plants is basically it's stored in the form of starch. In animals, however, energy storage is in glycogen. So bottom line is these are polysaccharides that are made up of a whole bunch of glucoses, right? So in our bodies, we store glucose in the form of glycogen, okay? If our blood glucose levels drop and we need more glucose, right, to provide energy for our cells, basically we'll break down glycogen so that we have more glucose available, okay? And so the same is kind of true for plants with starch. All right, so these are storage molecules, meaning there's lots, that's where glucose is stored. It's stored in these more complex carbohydrates. All right, so these are 2 polysaccharides that, again, are involved in energy storage. Starch in plants, right? Glycogen in animals. If we talk about structural polysaccharides, and I kind of alluded to this before, 1, cellulose, okay? Cellulose is found in the cell walls of plants. Okay, it's a structural polysaccharide or a structural carbohydrate. So it in fact is the most abundant carbohydrate, okay, and the most abundant organic molecule on Earth. Okay, because if we think about how many plants there are, all of their cell walls are composed of cellulose. Chitin is another type of structural carbohydrate, and it's found in the cell walls of fungi and also in the exoskeleton of animals, right? So if you think about an insect exoskeleton, it's composed of chitin, okay? Chitin has antibacterial and antiviral properties, and actually it's used in wound dressing and suture material because of those antibacterial and antiviral properties, but also because it's structural and it's a strong carbohydrate. Another one, peptidoglycan, another structural carbohydrate, a structural polysaccharide, is found in the cell walls of bacteria. All right, so this is just showing you, the structures of starch. So you can see here, showing you a potato. We always think of potatoes as being starchy, right? And actually, if you look microscopically at the potato, you can actually see starch granules, okay? And so this is actually showing you the starch, right? The structure of starch. So this is actually showing you here one form where it's non-branched and another form where it's branched. Nobody's going to be responsible for identifying these molecules based on structures. But again, it is made up of all glucoses, okay? Here, this is showing you glycogen, okay? You can see how the structure is, different than starch. This is actually showing you cellulose and kind of showing you the structure of cellulose, and you can kind of see, think about like celery, right? How it's stringy. So if you think about the structure of this carbohydrate, it's more has fibers, right? Fibrous, and so that's because of the way that these molecules are formed. Okay. All right, and this is just showing you here, plants' cell walls contain cellulose. I mentioned that the exoskeletons of some animals like insects, also crustaceans, contains chitin. And then another type of polysaccharide called peptidoglycan you'll find in the cell walls of bacteria. All right, so we move on. Next group of biomolecules that we're going to talk about is lipids, okay? And so it says lipids are varied in structure, right? They're non-polar, they're large, so if they're non-polar, they're insoluble in water. If we think about another term to describe that, it's hydrophobic, okay? In terms of function, a bunch of functions here, right? Long-term energy storage. So if we think about fats in our body, right? We can store energy in the form of fat. The body can then break down fat to then ultimately get ATP as well, okay? Structural components, membrane. Plasma membrane of all your cells is made up of phospholipids, okay? Special type of lipids. We'll talk about them. Involved in cell communication and regulation. So steroids or steroid hormones like testosterone, estrogen, those are lipids, okay? And so when we say about cell communication and regulation, we're talking about sort of hormonal regulation. Think of it that way, okay? Protection. If we think about fat, protecting our organs, heat retention, same thing. If we think about subcutaneous fat underneath our skin, it's going to insulate us and help us retain heat. Okay, so some varieties. I already mentioned fats. I mentioned phospholipids. I mentioned steroids, right? Fats, oils, phospholipids, steroids, and waxes are all different varieties of lipids. Okay? All right, so in terms of looking at some of those different varieties, we can see some of the different functions here, right? So fats, long-term energy storage, and insulation in animals. We already talked about that. If you're talking about human uses, things like butter, lard, oils, long-term energy storage in plants and their seeds. For us, we use them as cooking oils, right? I mean, you can have safflower oil, olive oil, vegetable oil, okay? Phospholipids, components of plasma membrane. There is no sort of human use for that. Steroids, okay? I mentioned sex hormones like estrogen and testosterone. Also, cholesterol is a steroid. It is a component of the plasma membrane. Also, we know cholesterol is also part of a, how do I want to, I guess, put this, as sort of a complex of proteins where basically it's carried around the body, right? In this sort of lipid protein carrier, but it could lead to or is involved in kind of right, heart disease and atherosclerosis, right? The sort of accumulation of lipids in the arteries. Okay. Says here they're used as medicines. Waxes, right? Protection, prevent water loss. If you think about the surface of a plant and how a leaf feels, it has a waxy cuticle, right? That protects from water loss or prevents water loss. Beeswax, earwax, and obviously we sort of exploit these in polishes and also in candles. Okay, so let's talk about fats and oils first. So another word or another name for fats is triglycerides, okay? And that basically describes what makes up fats, okay? So involved in long-term energy storage, right? What do these triglycerides or fats consist of? One glycerol molecule, right? Linked to 3 fatty acids, again, by dehydration synthesis, okay? So whenever we're talking about joining subunits together, the reaction that occurs is a dehydration reaction, okay? Fatty acids are either unsaturated or saturated. I started off part 1, sort of introduced this chapter talking about trans fats a little bit. But anyway, so this is kind of we're sort of coming back to that. So if we talk about an unsaturated fat, right, it's composed of these unsaturated fatty acids, okay? So unsaturated fats tend to be liquid at room temperature, things like olive oil, right? And these have one or more double bonds between the carbons. So an unsaturated fat has double bonds in the structure, and that makes more space, okay? And that's why we see that these are liquids at room temperature, right? And so that's what this is saying here. The double bond prevents close packing between the fatty acids. And I'll show you a picture that illustrates that in a second. Saturated fats, on the other hand, do not have any double bonds, okay? It's all single bonds. They are solids at room temperature, things like butter and lard. And because they have no double bonds, those fatty acid chains are really tightly packed, okay? If we talk about a trans fatty acid or a trans fat, there is at least one bond that's in a trans configuration. Okay. And so I only see some. Yeah. So I was going to say, we'll talk about trans fats a little bit more. I think I added that slide in because I think the book kind of didn't talk about that, just to kind of clarify. All right. So if you look here, right, this is what we were just talking about. We talked about unsaturated versus saturated. Okay, so let's kind of just do that first, just because I just ended with that. This is showing you, right, think about plant oils, corn oil, olive oil. And you can see if you look at this unsaturated fatty acid, right, so remember these are the fatty acids. So if we're talking about a fat or a triglyceride, it's glycerol plus 3 fatty acids. Okay, and remember these are joined together by, I don't know why that's doing that, joined together by a dehydration reaction. If we're talking about breaking down, it's hydrolysis, okay? Same reactions we talked about before. All right, so coming back to this, if we talk about an unsaturated fatty acid, you can see here's an unsaturated fatty acid, right? Part of this fat molecule of this triglyceride. It has double bonds. Okay, and so if you look at the difference between this and this, you can see these double bonds create space. Okay, and if you look at this space filling model here, you can see there's space between these fatty acid chains, whereas there's no space here. Saturated fat, solid at room temperature. Unsaturated fat, lots of space, double bonds, liquid at room temperature. Okay, we should be able to tell the difference between these. I would never ask you, to name this fatty acid, but if I showed you this and asked you if it's unsaturated versus saturated, we should be able to answer that question. Okay. All right. Just hold on one second. Okay. So again, just to focus on that top part that I sort of already went through, you can see, again, if we're talking about the formation of a triglyceride or a fat, right? We have a dehydration reaction to join the glycerol to these fatty acids, right? And then, and you can see there's 3, right? We're joining here, here, and here. So that's where this 3 comes from. And then if we're talking about breaking down this triglyceride, right? We're talking about a hydrolysis reaction. Same reaction as we talked about before. All right. The one thing that I, again, just want to sort of clarify, these trans fats. The trans configuration just means you have these, you sort of have a different configuration around this double bond here. Okay. So this is showing you an unsaturated fat. This is a trans fat. So basically, if we talk about a hydrogenated oil, right, that's something that you see, or you see packages that say no hydrogenated oils. These are basically synthetic conversion of saturated fats. They're basically converting. It's sort of artificially, not artificially, but synthetically, I guess is the right word there, converting things to make them hydrogenated, right? By adding hydrogens. Okay, so things like peanut butter and margarine that basically have this really good shelf life. So margarine is vegetable oil, but basically you made it into this hydrogenated oil so that it increases the shelf life. Okay, so it says here the side effect of hydrogenation is that you get these unsaturated fats with these trans double bonds, okay? And that's where that word trans fat comes from, okay? And these trans fats contribute to atherosclerosis, which basically means, the sort of common term, hardening or thickening of the arteries, but it's accumulation of essentially of lipids or fats within in the artery wall that then will occlude that artery, right, and can lead to heart disease. But so basically, you're basically taking you're adding hydrogens to it, sort of synthetically, but as a byproduct of that, right, of this sort of side effect of hydrogenation, which just means adding hydrogens, you get these weird trans bonds and these trans fats. Okay, and it's believed that these trans fats are particularly more dangerous than saturated fats themselves. Okay, so remember, saturated fats don't have any double bonds. Unsaturated fat, double bonds. You get these trans fats when you sort of artificially hydrogenate these unsaturated fats to increase the shelf life. Okay, so again, think about something like margarine. It's vegetable oil, but it's been hydrogenated, and then you can end up with these trans fats. All right, moving on to another type of lipid. Okay, so that was all about fats. We were talking about fats or triglycerides. The next part talks about phospholipids. Super important class of lipids found in your membranes, okay, in your cell membranes. The structure is similar to triglycerides. And basically, it's one glycerol molecule linked to two fatty acids and also a phosphate group. And that's where the name phospho comes from, right? It says the fatty acids. Okay, so what ends up happening here is that part of this molecule is polar and part of it is nonpolar. Okay, so the fatty acids are nonpolar and therefore hydrophobic, right? We talked about this before. The phosphate group is polar and therefore hydrophilic, okay? And again, these will form plasma membranes. And really, they form plasma membranes just based on this chemical structure here, okay? Remember that if we think about our cells, we think about our body, they are in an aqueous environment, an environment that has lots of water, okay? So when these phospholipids are in water, like it says here, they'll aggregate to form a lipid bilayer. That lipid bilayer makes up your plasma membranes of your cells. Okay, and those polar phosphate heads will orient towards the water. The nonpolar fatty acid tails will be oriented away from the water. Okay, again, this is just solely based on the chemical properties which are outlined here. Right, so let's look at a picture here. Right, so that's what we were just referring to. Right, if you put these in water and if you look at the plasma membrane, there's lots of water here, there's lots of water here. These are the polar phosphate heads. These are hydrophilic. They face towards the water. These fatty acid tails, which are hydrophobic, face towards the inside and form that bilayer because they do not like water and they're sort of turning away from the water. All right. This is just basically showing you the structure of that phospholipid. Here's your fatty acids, two fatty acid tails, right? Here's your glycerol and the phosphate group here. All right, another type of lipid that I already mentioned, steroids. Okay, so these are composed of 4 fused carbon rings. Again, we don't need to draw, structures of any sort. And then you have these fused rings, but then there's also functional groups that are attached to those carbon rings. In terms of function, component of animal cell membrane, that's cholesterol as part of the cell membrane as well. Also regulation if we're talking about steroid hormones like testosterone and estrogen, okay? Also, I said cholesterol is present in the membranes. It is also a precursor, meaning it's necessary to make other steroids and other steroid hormones like testosterone and estrogen. So if you don't have enough cholesterol, if cholesterol is not there, your body will not be able to synthesize testosterone or estrogen, okay? So this is just showing you here again, this is what I was just telling you, cholesterol is the precursor, meaning testosterone and estrogen are derived from cholesterol. If this cholesterol wasn't around, you can't make testosterone or estrogen. And so this is just showing you peacock here, right? So this is the male and this is the female here. And so showing you presence of testosterone versus estrogen. Okay. Another thing worth mentioning, and I sort of kind of alluded to this too a couple slides ago, in terms of cholesterol, right? Probably when we think of cholesterol, this is what we think of, right? We think of blood cholesterol levels. Maybe we think about high density or HDL versus LDL, right? If you go get your cholesterol tested, you'll get a total cholesterol reading, but really what's important is to look at the breakdown, right? In terms of the ratio of HDL, high density lipoprotein, versus LDL. Okay, so the HDL is the good cholesterol, the LDL is the bad. And so again, this LDL, low density lipoprotein, it's basically showing you how, it's referring to how cholesterol is carried around in the blood. Right, so you see this here, the orangey thing here is the cholesterol, and then these are a whole bunch of other proteins that are sort of part of this complex here, okay? Where again, here's your cholesterol, and this is the part. So that's what this is referring to in terms of the HDL versus the LDL, the lipoproteins that essentially carried around. Okay, so that's what this is telling you here. And so if you look here, this is showing you a normal artery, and then this is an artery with this plaque buildup. Okay, so again, LDL is responsible for delivering cholesterol to the cells, right, for membrane production. And that LDL has a tendency, if there's too much, to form plaques in the arteries. On the flip side, HDL actually can scavenge excess cholesterol and basically return it to the liver to be broken down and basically destroyed, right? So that it doesn't accumulate. And so that's why we call the HDL the good cholesterol and the LDL the bad cholesterol. Okay, so like I said kind of before, increased ratio of LDL, meaning more LDL versus HDL, it leads to high risk for heart disease because this will happen. Okay, all right. Another type of lipid, waxes. Okay, so these are long chain fatty acids that are bonded to a long chain alcohol. They are solid at room temperature. Again, think about wax, right? Solid at room temperature. If you heat it, it melts. They're waterproof, resistance to degradation. Really, in terms of functions, it's protection. Think about earwax, think about plant cuticle, beeswax. These are all protective, right? And so this is what this is showing you here. You have your long chain fatty acid and the long chain alcohol, okay? Which is basically these are bonded together, right? Okay, so this is just showing you again this sort of waxy substance. This is kind of surrounding the grapes, but also surrounding the leaves, and then also showing you beeswax. All right, we move on to the next class of biomolecules, proteins. Okay, so proteins are polymers of amino acids that are linked together by peptide bonds. Okay, so a peptide bond joins two amino acids together, right? Again, two or more amino acids joined together are called peptides, hence the name peptide bond. Long chains of amino acids joined together are called polypeptides. Okay, so when you have a lot of amino acids joined together, we call it a polypeptide, just like we called, things like starch, polysaccharides. All right, protein. Now the term protein is a polypeptide that has folded into a particular shape and then has function. Okay, so polypeptide is just the string of amino acids. It is not a protein until it properly folds. Okay, it will not function as a polypeptide. It needs to fold into a protein. All right, in terms of functions of proteins, there are many. Okay, very diverse set of biomolecules here. Here are some: metabolism, right? Enzymes. Most enzymes are proteins. Okay, enzymes will catalyze reactions, right? And if we think about metabolism, you can think about metabolism as being all the chemical reactions that occur within a cell. Support proteins like keratin and collagen, which are structural proteins. Okay, transport, hemoglobin, membrane proteins, defense, antibodies. Antibodies are proteins. Okay, we hear lots and lots about antibodies in this sort of age of COVID, right? Regulation, hormones. There are, I just mentioned steroid hormones, right? But there are hormones that are proteins, okay? Motion, muscle proteins, right? So microtubules as well, but muscle proteins like actin and myosin. All right, so we should know these functions of proteins and understand that they are pretty diverse in terms of functions. We said that proteins are made up of amino acids, right? The monomers of proteins are amino acids. There are 20 different amino acids. You do not need to memorize any of the amino acids. Maybe we just need to know one from each class. Okay, so I'm going to talk about that in a second. I'll show you a picture. But if you talk about an amino acid just in general, right? This is basically showing you something that's common between all amino acids, and then this R group is the rest of the molecule, meaning this R group will be different depending on which amino acid you're talking about. Okay, and so here's some examples, right? And so we can divide them up here in hydrophobic, hydrophilic, and ionized, right? Or acids here. So you should know one example from each one of these groups. Okay, so if I say name a hydrophobic amino acid or a nonpolar amino acid, valine, methionine, those are examples. Please do not try to draw structures or memorize structures or anything like that. Just know one example from each group, okay? Polar amino acids, hydrophilic, they are listed here: cysteine, serine, tyrosine, glutamine, okay? Ionized, right? Think of ones that have acid in their name: glutamic acid, aspartic acid, lysine, arginine, histidine. These are all examples of ones with ionized R groups. Okay, you can see here. Okay, these are ionized. So see this negative charge here. So anyway, just one example from each. All right, so if we talk about, again, synthesis and degradation, same thing, same reactions we've been talking about before. We're talking about polypeptides, we're talking about proteins, it's two amino acids joining together. So here you go, dehydration reaction, right? Peptide bond forms between these two amino acids. You lose water in the process, dehydration. If you go the other way, it's hydrolysis. This is just showing you a dipeptide, right? Again, for simplicity's sake, when we're talking about inside our cells or inside our bodies, usually many more amino acids, right? And in proteins. Anyway, hydrolysis reaction breaks it down, right? Degradation or breakdown, breaking this peptide bond and then having two separate amino acids again. Okay, now, as I said, a protein does not function until it is folded properly or has its proper shape. Okay, so a polypeptide now needs to fold. Okay, and so that's what it's saying here. And one thing to mention here is that proteins can, if they lose their shape, we call it denatured. Okay, so in terms of heat, a change in pH, those will disrupt protein structure. So if we think about, having a really high fever, a high increase in body temperature can start to denature proteins. You're in trouble if your proteins start to denature. We said almost all enzymes are proteins. And so if enzymes aren't able to function, none of those chemical reactions can function. And therefore it's going to severely disrupt the function of the cell, the function of the body. Okay. So 4 levels of structure here. So now we're talking about, you already have your polypeptide made. Now it needs to fold. Okay. So we sort of describe this in different levels: primary, secondary, tertiary, and quaternary, right? So primary is just the sequence of the amino acids. That's it. Secondary, you see things called alpha helices and beta pleated sheets, okay, which are held together by hydrogen bonds. Just think of this as kind of the initial sort of forming or initial folding, okay? Tertiary is the final overall 3D shape, okay? So more folding is occurring. And there's more complex interactions that occur, right? So it says hydrophobic interactions, hydrogen bonding, ionic bonding, covalent bonding, a whole bunch of different interactions that are occurring to fold that protein properly, okay? So the sort of, again, the initial sort of formation of these types of shapes, these alpha helices and beta pleated sheets, are part of secondary. Tertiary is that final overall folding, and it's more complex folding. Quaternary, you only have a quaternary structure if you have more than one polypeptide. So not all proteins have a quaternary structure. All proteins have a tertiary structure though. Okay, so let's look at a picture here. So here showing you the primary structure. It's just all amino acids joined together, a polypeptide chain, right? Just some peptide bonds. If we talk about secondary, it's the formation of alpha helices and beta pleated sheets, and that's really primarily caused by hydrogen interactions or formation of hydrogen bonds. Tertiary, you can see much more complex, much more folding that occurs. And then quaternary, if you have more than one polypeptide chain, they will then join together to form that more complex protein. All right, examples of fibrous proteins. Remember I mentioned in the beginning when we did functions here, right? Things like keratin and collagen. And so this is illustrating that. Okay, so some fibrous proteins. For example, this is illustrating keratin. Okay, so if you look at panel A there, okay, you find keratin in hair. And basically what this is showing you, the straight versus the curly hair here, is that if you get a perm, right, it will alter the secondary structure of keratin to form these kinks and to make the hair curly. okay? If we look at silk here, right, made by spiders, most of the keratin here is in beta pleated sheets, and you can see that structure. So the point is, I guess, the difference between these three pictures is differences in structure. It's the same protein, and we're kind of focusing on keratin here, but you can have differences in terms of the consistency, kind of, or of the hair, right, based on the secondary structure of keratin. Okay. All right. Now, worth noting is that there are diseases that are caused by misfolding of proteins. Okay. So we call these protein folding diseases. So I already told you this, that proteins can only function if they are in their proper folded state, in their correct shape. Okay. And there are actually a certain class or group of proteins called chaperone proteins that will help proteins fold into their shape. Okay, so if you have a problem with chaperone proteins in general, it's going to affect every other protein in the cell, right? And then a whole bunch of proteins wouldn't be able to be folded correctly. And so it says here, a new research indicates that they can correct misfolding of new proteins. The point is that chaperones help new proteins fold, okay? There are diseases, again, that are associated with misfolded proteins. So Alzheimer's disease is associated with misfolded proteins. And it may be due to, like I said before, chaperone proteins that aren't functioning properly. Okay, and so when proteins become misfolded in diseases like this, they then aggregate together, and then they can cause neurons, cells in the brain, to die, and obviously lead to dysfunction. Okay, so degeneration of the brain. Mad cow disease is a disease that's actually caused by something called a prion. Okay, so a prion is this kind of weird. It's not a virus. It's like a protein, or it's not really living itself anyway. This sort of weird thing that will cause other proteins to misfold. Okay, so we talk about prions later on a little bit more. All right, last class of biomolecules to talk about, right, are nucleic acids, and we're just about done. Nucleic acids are polymers of nucleotides, okay? So nucleotides make up nucleic acids. The two types of nucleic acids that exist, that we talk about, right, in biology are DNA, deoxyribonucleic acid, and RNA, ribonucleic acid, okay? DNA, genetic material, okay? Obviously, if we talk about our genes, made up of DNA. If we talk about RNA is involved in protein synthesis and regulation of gene expression. So DNA and RNA really go hand in hand. But if we talk about our genes and the genetic information, right, that or the storage of genetic information, it's in the DNA. Okay. All right. So we want to talk about a nucleotide first, because remember, the nucleotides are the subunits or the monomers of nucleic acids like DNA and RNA. Okay, so each nucleotide has three parts: a phosphate group, a pentose sugar, and a nitrogen-containing base or nitrogenous base. Okay, if we talk about the types of nucleotides, you probably have heard of these before. In DNA, we have adenine, guanine, cytosine, and thymine. In RNA, all of the same, except that there is no thymine. It is uracil. Okay, so and if we talk about pairing of these nucleotides, right? Adenine pairs with thymine, guanine pairs with cytosine. Okay, in RNA, adenine pairs with uracil, guanine with cytosine. All right, again, same reaction. When we join nucleotides together, it's a dehydration reaction. Okay, here you go. Here's your. This is showing you nucleotide structure, right? Here's your nucleotide. Okay, here's your nitrogen-containing base. Here's your entire nucleotide. This is showing you deoxyribose versus ribose. Okay, it's the difference. It's a five carbon sugar here, but you can see you have one less oxygen. Okay. If we also talk about these nucleotides, right, we can divide them up into two groups called pyrimidines and purines. If you notice, purines have a double ring structure, protamines only a single ring, okay? And so cytosine, thymine, and uracil are all pyridamines, purines, adenine, and guanine. Okay, so we should be familiar with that. All right. And I'm not going to show you a specific reaction in terms of joining them together, but they are going to join together by dehydration synthesis, and they're going to be broken down by hydrolysis, just like we talked about before, and they form a linear molecule or a strand, right? So if we look here, right, we have these nucleotides joining together here, basically to form, you know, the polymer, which in this case is showing you RNA, okay? So let me just go back here in terms of the structure a little bit more. I probably said most of these before. If you talk about the backbone, right, of the nucleic acid here, right, it's alternating phosphate and sugar molecules, sugar phosphate backbone. That's what this is saying. RNA is single-stranded, DNA is double-stranded, okay? And you can see here, right, in RNA single-stranded, and you can see the nitrogenous bases facing in towards the center here, okay? It says DNA is composed of two strands held together by hydrogen bonds between the nitrogen-containing bases, right? And those two strands will twist around each other to form a double helix. I already said this. Adenine bonds with thymine, cytosine with guanine, okay? We call this complementary base pairing, okay? Adenine doesn't sometimes pair with thymine. Adenine always pairs with thymine. Cytosine always pairs with guanine. All right, so if we look at DNA, you can see here, right, between these nitrogenous bases, you have these hydrogen bonds that form to join them together. And then you can see this double helix structure. Again, what's here in aqua is the sugar phosphate backbone. What's facing in towards the middle are those nitrogenous bases, okay? And this is, again, showing this complementary base pairing, cytosine and guanine. If you notice, 3 hydrogen bonds here, only 2 hydrogen bonds between adenine and thymine, okay? Right, so a purine is always going to pair with a protamine. All right, and so just to kind of compare DNA versus RNA, I probably said all of these already. In DNA, the sugar that forms that sugar phosphate backbone is deoxyribose. In RNA, it's ribose. The bases that make up DNA, right, and the bases that make up RNA. DNA double-stranded, RNA is single-stranded. DNA forms a helix, RNA does not. All right, one last thing to mention is a special nucleotide that's ridiculously important to the cell because it is the energy currency of the cell, ATP, okay? ATP is adenosine triphosphate. It's composed of adenine, ribose, and 3 phosphates, okay? It is a high energy molecule because those last 2 phosphate bonds are very unstable and they contain a lot of potential energy. So when you break that phosphate bond, it liberates energy, okay? So it says here, hydrolysis of the terminal phosphate, if you're talking about ATP, 3 phosphates. If you break one of those bonds and break off one of the phosphates, you will then end up with ADP diphosphate, right? And then a free phosphate, but then you're also going to liberate energy, and that energy is used to do work within the cell. Okay, I already said this. All right, so this is showing you that here. Here you go. Here's your ATP triphosphate, right? And basically, if you cleave off, you break this bond here, right? We're breaking that bond. You can see that now you end up with ADP and a free phosphate, but you also end up with lots and lots of energy. So when the cell needs energy, it will cleave that phosphate group from ATP. So it will convert ATP to ADP, and that will liberate tons of energy that's used by the cell to do work, okay? We'll talk about this again in the subsequent chapter, but remember, it is a nucleotide, okay? Special nucleotide ATP. All right, and that concludes part 2 of chapter 3.